Hi, today we will be discussing plant germplasm conservation. We know green plants how important it is for our existence. The green plants are the primary producers of agro ecosystem. They are the primary species on our earth, and that green plants are endangered now due to many reasons. And to stop this onslaught, to stop this loss of green plants, germplasm, way back in 2002, a strategy called Global Strategy for Plant Germplasm Conservation was approved, and this is a very important strategy that paved way for our actions, the action towards conservation. To start with, we need to understand what crop germplasm means. It means the seeds, plants or plant parts that are useful in crop breeding, research or conservation. This crop germplasm, which encompasses all the plant parts, it is very important, essential resource for crop breeding activity, for research and for conservation also. For every information that has been encoded in the germplasm, that has to be used for the benefit of the mankind, for the humanity, and that is why it is a very important resource as far as the agriculture is concerned, as far as the humanity is concerned. And why and how, what for this crop germplasm is needed for our mankind. It is a very important resource. I just told you it is benefiting to mankind, and also it helps in preserving the environment. The plants, it is having it is a survivor of gene pool. It has encountered the recombination breeding, it has encountered complex breeding processes, mutation, so on and so forth, and because of that thing it is a repository of genes, and this particular germplasm are available in the farmer's field as recombinants in the form of seeds. It is available in the herbarium, it is available elsewhere, but these particular resources are definitely useful for our crop improvement activity, plant breeding activity, and this forms an important repository of wonderful, interesting, qualitative genes, and these genes are very essential for improving the crops. And as far as the crops are concerned, we know fully well that how the cultivated crops what we are having now, it has been improved when you compare with the old wild species. A lot of change, a sea change in fact, that has taken place between a wild species and the present day cultivated crops. So that is made possible because of the valuable genes that is present in the germplasm, and that is made possible because of the selection process, that is made possible because of the natural or artificial selection, and everything the credit goes to the germplasm and of course the natural processes. And if you look at it, the germplasm is required for developing new crops to meet the needs of the mankind, for medicinal purposes, industrial uses, as pest repellents or nutritious foods. When you look at it, the research on plant being done for finding out the constituents of the plants that can help in managing the diseases like cancer, many diseases. We are looking for the medicinal properties present in the plants. So from there we can manage to develop a new crop plants, new constituents, new medicine that is possible, and we are looking for it, and also for developing a fiber crop, annual fiber crop, short duration fiber crop. So our fiber demand can be met by developing such kind of crops, and also by going for qualitative foods, nutraceuticals, by going for biofuels, by going for a crop which can harvest the best possible solar energy in an efficient manner, that is also best thing, and also by looking for a crop which can emulate the biological mechanisms, pest management mechanisms to other crops. So all the things we are moving for developing the new crops, that is coming from the crop germplasm. The crop germplasm is a very essential component because it meets our every need, mankind's need, and when you look at it, is this germplasm at no cost, we should not miss it, we should not lose it. And as far as the plants are concerned, more than 2,50,000 angiosperm plants that has been described, and within that thing less than 500 species have been used for human use, and within that 500 species also only 30 species meets the 95% of the calorific demand. So, how we are narrowing our focus, we are dependent only on few crop plants, that only 30 species. See, and also very importantly, this crop plants whatever we are talking about, they are cultivated varieties. These cultivated varieties are having a narrow genetic base, and because of this narrow genetic base they are prone for vulnerability, they are vulnerable, genetic vulnerability to biotic stresses, abiotic stresses. So, now coming to the sources of crop germplasm, the first and foremost important source is land races. These land races have been developed in the agricultural fields by the farmers, by the pre-agricultural societies. They have developed this land races. For developing this land races, perhaps it would have taken more than 1000 years. These land races are a repository of wonderful superior genes. Why? Because these land races are in a position to adapt itself to the local condition, local environmental conditions. They have all the what you call the genes to manage the stresses, either the biotic or abiotic stresses. They are having all the possible things, but the yield of this land races is low, but the yield is stable. So, we need to look for it, we need to conserve this land races, because land races having a lot of potential for plant breeding. Next important thing as far as sources are concerned, obsolete varieties. These obsolete varieties, unlike the land races, these obsolete varieties have been developed by scientific plant breeding activity, and these obsolete varieties once were cultivated, but now they are not cultivated due to certain reasons. Perhaps the varieties may not be performing, and also it may not be outperforming the cultivated varieties, the yield may be less. There are many varieties, NP varieties, K65 varieties of wheat that has been now obsolete. Like that, for every crop there are many obsolete varieties. Third important thing is the cultivated varieties, which are high yielding in nature. These high yielding nature varieties are very, very important. Why? Because they are responsive to the fertilizers, responsive to the irrigation, responsive to the human management. Because of these management, human management and agricultural inputs subsidized, because of that thing, these particular cultivated varieties are high yielding. They are semi dwarf in nature, they are photoperiod insensitive. So, there are so many things, the benefits that is accruing in cultivated varieties, that is making our agriculture production also, we have crossed higher yields, that is because of this high yielding cultivated varieties. This also forms an important part of crop germplasm. And next important thing as far as the sources of germplasm is concerned, they are breeding lines. When you look at it, the plant breeding activity, it takes a lot of time. In the process of plant breeding activity, many lines have been developed, and these breeding lines also repositories. When I say repositories, they are also having the germplasm, they are also having the beneficial parts, beneficial genetic material, and that also should be assembled for crop germplasm analysis. And last one is the wild forms and wild relatives. When I say wild forms and wild relatives, it is wild, it is thriving in the nature, but it forms an important source of crop germplasm. These wild forms, directly from these wild forms, cultivated crop plants have been developed. It can easily bred with the cultivated varieties, but on the other hand, the wild relatives are related to the cultivated varieties by descent. Maybe the ancestors are common, but these wild relatives cannot easily cross with the cultivated varieties, but both are very, very important thing. So, the wild forms and wild relatives are having lot of genes that provides resistance to the biotic stress and abiotic stresses also, and also it is having the genes for adaptation. Only when the crop plants are adapted, we can domesticate it, and only when we are domesticated, we can improve upon the domesticated variety to increase the yield. So, that is a very, very important thing, that is the part of this wild relatives, and we have used this thing, this wild forms and wild relatives for improving our crop varieties. So, many of our current high yielding varieties, in case of sorghum, maize, rice, wheat or many other crops, the increase in the yield or any other parameter for that matter, we have taken the genes from these wild forms, wild relatives, and these genes have been transferred to the cultivated variety. Just because of this thing, our present day cultivars are high yielding in nature. And not only the yield, the many parameters have been taken into account, the physiological efficiency. Look at the drought resistance, pest resistance, disease resistance, and many factors are been incorporated in the present day cultivated varieties because of the genes we have taken from the crop germplasm. We have looked for it. So that is the reason why we need to look for this crop germplasm, and we have to look for the sources also, and we have to conserve it also. And the conservation, before coming to the conservation, this germplasm are having a lot of variability, and the variability that is existing in the germplasm is due to mutation, is due to migration, it is due to recombination, it is due to genetic drift, it is due to these all factors combinedly were responsible for creating variation. The genetic variation, it's a very important aspect as far as germplasm is concerned. Only when there is a genetic variation, then only evolution will be possible, then only selection process will be possible. Selection, either it is a natural selection or artificial selection, that will be possible only when there is a genetic variation. And this genetic variation I talked about, it is a product of many factors. The prime factor is mutation. The prime factor is mutation. It is a very important factor that is caused, and that is because of that reason many of the superior genes have been conserved in the wild varieties, wild cultivars. Now coming to the germplasm conservation, the management of human use of the biosphere so that it may yield sustainable benefit to present generations while maintaining its potential to meet the needs and aspirations of future generation. This germplasm conservation is a very important thing. We need to conserve it at any cost, definitely, otherwise if we lose it, it is irreplaceable, we cannot get it back. And because we are dependent on this thing, we will be dependent on this particular germplasm, because the kind of resources that is existing in the germplasm we do not know. We need to understand that particular thing, we need to scientifically understand that what is present in this thing. So for that a lot of research has to be done. So in nutshell, what I want to say is that this germplasm is a very important, germplasm conservation is a very important exercise as far as the sustainable management of biodiversity is concerned. And how to do it? There are a lot of approaches, and when you talk about the approaches and what is the need for it, what is driving, the reason is the genetic diversity. We are losing it because of many factors: prime genetic drift, genetic wipeout, genetic erosion. Because of this, what happened, we are losing the genetic diversity. At any cost we have to conserve it. And how to do it? There are a lot of approaches. We back in 1970s, Food and Agriculture Organization woke up for the call. Definitely it is a wake up call. Why? Because they were threatened, the organization, scientific community was threatened, because they were sure that the genetic diversity, we are losing it because of many reasons: human action, because of a lot of change in the environment, lot of change in the even climate also. So many environmental factors are changing, the global change that is taking place. So that is all going to cause a stress to the genetic diversity. So they came forward with a strategy, and that strategy they wanted to discuss time and again, and this discussion went further in the United Nations Conference on Human and Environment, 1972 Stockholm Conference. In that conference also they discussed in detail about the strategies, about the approaches to conservation, and the outcome of the conference, or the outcome of the strategy, outcome of all the discussions was simple. They said that there are two strategies, that is, one is ex situ conservation, another one is the in situ conservation. Both are very important thing, both are having complementary nature, both has to be taken into account in unison, then only we can conserve the valuable genetic resources, valuable crop germplasm. First and foremost important thing as far as the conservation strategy is concerned, that is in situ conservation. The in situ conservation, it means that the continued maintenance of a plant population within its ecosystem to which it is adapted. We need to maintain, we have to conserve the germplasm in its own habitat. That is a very important exercise, and it is a very important strategy also, in situ conservation, because it is having a potential to conserve the crop species, it is having the potential to conserve the wild relatives, it is having the potential to conserve the distant relatives also, crop plants also, cultivated plants also. That is how the key this strategy in situ conservation, we need to focus on that particular thing. So the potential is huge, and definitely we need to look for this strategy and implement it appropriately. And why it is so important? Because this in situ conservation strategy allows natural forces of evolution to take place, genetic shifts to take place. Because of the natural forces of evolution, what is taking place? Because of this natural forces of evolution, what happened? Only the fittest can survive. The genetic resources, the plants, they are under selection process. When I say selective process, what happened? The fittest can survive, survival of fittest and finest. They have all the valuable genes under that particular condition, under that particular environmental condition, only those plants can survive. So in other words, the in situ conservation helps us to have an evolutionary process. A new crop evolution will be possible. Evolution is not stopped, is not ceased in case of in situ conservation. And it also adopts an important strategy that is called system approach. When I say system approach, the conservation strategy, the plant population is conserved in its own ecosystem, along with this abiotic components, it has been conserved, and because of that reason many forces, many processes are taking place simultaneously. The crop plants, they get the benefit of mutualism, they get the benefit of co-evolution, co-adaptation, and because of that reason what happened, the processes like mutation, recombination, that is also taking place. Because of these all the things that is taking place in the system, the best is we are getting evolved species. Evolved species, when I say evolved species, that is a product of natural evolution, and this is how the in situ conservation is important. And also if you look at it, the in situ conservation has furthermore many features, but to highlight it, this particular thing, the system approach has created definitely a hallmark in situ conservation strategies. And the advantages, when you look at it, the prime advantage, as I told you, it conserves a wide range of interesting alleles, qualitative alleles, everything is present in this thing, and that is conserved by adopting in situ conservation. And very next important thing as far as the in situ conservation is concerned, it is suitable to species, plant species requiring natural habitat. Certain crop species require natural habitat for conservation. For example, in a climax ecosystem, moist deciduous forest, the tree plants require or dependent on other species for its survival. So in that case we cannot think about other strategies, we have to look only for the in situ conservation. We have to conserve those crop plants or only under natural habitat only. So for those ecosystem, the in situ conservation is the appropriate strategy. Next example, if you look at it, certain species may have dormancy problem. When I say dormancy is a very important thing. So this dormancy cannot be removed by artificial means, only by natural means this dormancy can be removed. So those species can be conserved by adopting in situ conservation. There are certain other species also. So they have a unique breeding mechanisms, and the unique breeding mechanism means that they may be pollinating by a unique butterfly species or a bird or a bat. So under this condition we cannot think about another strategy, we have to adopt in situ conservation only, then only those species can be conserved. It is uniqueness for the species uniqueness. And next important thing as far as the in situ conservation is concerned, it aids in the conservation of disease and pest resistant species. I told you about the natural evolution, that is all because of that thing. The land races, the relatives, they are repository of the genes for resistance, pest or diseases. So that all that these species are conserved because of in situ conservation, and this in situ conservation also forms an important platform for research. The research can be done on the species in their natural habitat. The species can be examined for its resistance against the drought, for resistance against the flood condition, for resistance against many other factors, biotic or abiotic. So that is possible. This in situ conservation provides the pathway for the research on the species in their own habitat. That is a very important thing. And next important thing is as far as the advantages is concerned, on farm conservation. When I say on farm conservation, it is a very unique strategy for the conservation of biodiversity, agricultural resources. The agricultural resources can be conserved by adopting in situ conservation. It is called as on farm conservation. And because of this strategy, the wild relatives, the weedy plants and many other relatives also, the crop relatives, all are conserved, and that leads to what? A kind of, the ecosystem will be enriched, the ecosystem resilience will be enriched, because the resilience capacity of the agro ecosystem will be improved because of this on farm conservation. Because not only the crop plants, not only the wheat plants, every other species have been conserved. That gives an edge, that gives the existence for a sustainability of the ecosystem. That is the reason why resilience is increasing in case of on farm conservation. And also a kind of a dynamic and adaptation strategy that is also existing in on farm conservation. When I say dynamic adaptation, means that the crop plant or the plant, it will have an adaptation, it will have an co-adaptation, co-evolution with other species. It can be a microorganism, it can be a pathogen. Because of that thing, what happened, there is a development of pest resistant species or a disease resistant species that is possible, and that is this dynamic adaptation is a hallmark of on farm conservation. This on farm conservation is a very important aspect as far as the agrobiodiversity management is concerned. But this on farm conservation, the success depends on the active participation of the farmers. They are the prime stakeholders. They should be aware about the benefit of germplasm. They should be aware about the how this germplasm can be used for their welfare, for their benefit. And on the other hand, ex situ conservation is a very important thing. Like in situ conservation, be aware about the benefits, and also this on farm conservation will be successful when the extension activity, the transfer of technology, the incentives, the land rights, everything should be operating in unison, and only then this on farm conservation will be successful, and that is a very important thing for agrobiodiversity management. This ex situ conservation, it means that the conservation outside the natural habitat. The first and foremost ex situ conservation strategy is seed gene banks. In this case, the genes banks, seed gene banks, the seeds are conserved at sub-freezing temperature. Perhaps it will be minus 20 degrees centigrade, and this is an important strategy for conserving seed propagated plants. The seeds can be either the orthodox seeds or recalcitrant seed. The main difference between the orthodox seed and recalcitrant seed is the orthodox seed can survive even under low moisture content, but recalcitrant seed cannot survive under low moisture content. So the strategy will be different for conserving an orthodox seed and a recalcitrant seed. And in case of seed banks and at the genetic resource centers, there are two type of collection that is existing. That is, one is the base collection and the one is the active collection. The base collection, in this base collection, the seed accessions, they are kept for a long term, they are kept for a long term storage, more than 20 years, and under a very low moisture content and also under very low temperature. Perhaps it will be minus 20 degrees centigrade. But as far as the active collections are concerned, they are focusing on accessions, but these accessions are stored for only for 5 to 20 years. And not only that thing, these active collections are stored under the temperature also, it is more than 0 degrees centigrade, it is not less than 0 degrees centigrade, it is more than 0 degrees centigrade. And what is the important criteria is that the seed viability should not be less than 65%. If it is saved for 20 years, the seed viability should not reduce. That is a very important aspect of active collections. Both passive and active collections are present in the genetic resource centers. And next important thing is the cryobanks. The cryobanks, in these cryobanks, the cryopreservation of orthodox and recalcitrant seed species, pollen, embryos, embryonic axis, everything has been cryopreserved. And this plant material, whether it is a seed or a pollen or an embryos, it have been conserved at a very low temperature at minus 165 to 196 degrees centigrade, which is made possible by liquid or vapor nitrogen. This only can give such a low temperature. Under that low temperature, cryopreservation is taking place. Next important strategy is in vitro banks, in vitro tissue culture banks, wherein the fruit crops, musa, apple, rootstock and kiwi fruits and spice crops like curcuma, ginger, more than the Gingiber officinale, tuberous crops, sweet potato, diascorea, medicinal plants, many crop plants have been conserved by using the in vitro tissue culture banks, and wherein the temperature is ranging between 4 to 25 degrees centigrade. And next important strategy as far as ex situ conservation is concerned, it is the field gene banks. It is otherwise called as clonal collection, wherein the fruits and ornamental tree crops are conserved, and it is a live collection. It is a live collection, and the main focus here is the vegetative propagation. Why? Because of adopting this particular vegetative propagation, the heterozygosity can be maintained and true to type nature, and that is a very important thing as far as the field gene banks are concerned. And in India, if you look at it, there are many places where this field gene banks are existing. Jodhpur for arid fruits, Simla for temperate fruits, Thrissur, Akola, and for agroforestry crops also Jhansi is a very important place. And another one is the community gene banks, wherein the non-governmental organization like MS Swaminathan Research Foundation, Navdanya, Beach Bachao and many other organizations are involved in the community gene banks, wherein the main aspect is the creating awareness, and second aspect as far as MS Research Foundation is concerned, it is also having the medium term storage facility. And last but not the least, the strategy of ex situ conservation is the national herbarium that is existing in India at the Delhi NBBGR, National Bureau of Plant Genetic Resources, wherein the herbarium is having a collection of 14,000 specimens. It is a very huge collection. That is a very important aspect. And to sum it up, this crop germplasm provides immeasurable benefits to the mankind. At no cost we should not lose this invaluable resource, and this germplasm forms a foundation for scientific plant breeding. And last, the strategies, either it is a static or dynamic conservation strategy, both must be implemented efficiently. Only then this biotic resources or genetic resources can be used for the present as well as the future generations, and it will be forever. It will be definitely for the humanity, for the benefit of the humanity. Thank you.